Thank you for your interest in the Precision Immunology App-a-thon on COVID-19. This App-a-thon runs from November 30th, 2020 until January 29th, 2021 and comprises two tracks, a data science track for those who do not possess immunology knowledge, and a computational immunologist or CI track for those who are advanced bioinformaticians with immunology experience. The purpose of this video is to introduce you to the concepts of AIR and AIR-seq, familiarize you with the iReceptor gateway, and finally to do a walkthrough of the provided datasets for this App-a-thon. Let's start with a brief overview of T and B cells and their receptors. T cells are a type of white blood cell derived from bone marrow that develop into several distinct types once they migrate to the thymus gland. T cell types have many different immune-related functions. CD8 T cells, also known as killer T cells, are able to directly kill virus-infected cells as well as cancer cells. CD4 T cells, or helper T cells, function by indirectly killing cells identified as foreign. They determine if and how other parts of the immune system respond to a specific perceived threat. Regulatory T cells provide a mechanism whereby immune cells are able to distinguish invading cells from self. T cell receptors, or TCRs as I'll sometimes refer to them here, are found on the surface of T cells and are responsible for recognizing fragments of antigen as peptides bound to major histocompatibility complex molecules. TCRs are embedded in the membrane of the T cell and composed of two different proteins, an alpha chain and a beta chain, encoded by the TRA and TRB genes respectively. When the TCR engages with the antigen, it stimulates the T cell to become a CD8 or CD4 T cell. B cells are a type of white blood cell also derived from bone marrow that function by binding to an antigen, receiving chemical signal from helper T cells, and differentiating into a plasma cell that secretes large amounts of antibodies. The B cell receptors, or BCRs, are cell membrane proteins on the surface of a B cell composed of immunoglobulin molecules. Through biochemical signaling and by physically acquiring antigen, the BCR controls the activation of a B cell. Now with a bit of background, it should be easier to understand the concept of AIR. The acronym AIRR stands for Adaptive Immune Receptor Repertoire, which is the collection of antibody B cell receptors and T cell receptors that constitute the adaptive immunity of an organism, capable of recognizing a vast array of antigens including pathogens, autoantigens, allergens, toxins, and tumors. AIR-seq refers to the profiling of AIR with high-throughput DNA and RNA sequencing. The adaptive immune system is incredibly diverse. It has been estimated that there are upwards of 10 to the power of 13 human B cell receptor sequences. Quantifying and understanding this complex system is critical to vaccine development, finding biomarkers and therapeutics for autoimmune diseases, and in monitoring and predicting efficacy of modern anti-cancer immunotherapies. The AIR community develops guidelines and best practices for the generation, use, and sharing of AIR-seq data, and to address challenges involved in the management and analysis of these large data sets. The figure you see before you will help you to better visualize AIR-seq. Here we see B cells extracted from an infected individual and associated B cell response to infection, leading to expansion of antibodies present in the plasma. This individual's B cell repertoire can be interrogated by sequencing the variable genes of the antibody. Subsequent bioinformatic analysis of the sequencing data produces the type of data you are provided as part of this App-a-thon. So now that you're familiar with the concept of AIR, let's move on and introduce the iReceptor gateway. The purpose of iReceptor is to facilitate the curation, analysis, and sharing of antibody B cell receptor and T cell receptor repertoires for multiple labs and institutions. For this App-a-thon, the overall dataset provided by the Precision FDA team is composed of two datasets queried from the iReceptor gateway. DS track participants will only be required to utilize the provided dataset to create their tool, whereas CI track participants are required to use the provided datasets, including expressed sequences, as well as additional datasets either queried from iReceptor or identified by you. If you are participating in the CI track, you will want to request a user account so that you may perform queries and download additional datasets from iReceptor. In order to obtain a user account, you can email support@ireceptor.org, and you will be sent a username and password. Once you've obtained an account, you can start exploring and querying the data. The next portion of the video is primarily aimed at participants of the CI track. But those participating in the DS track may also find it informative to understand how we obtain the provided dataset. So let's walk through the iReceptor Gateway queries focusing on COVID-19 data. After you log in, you'll see this screen, which will show you a breakdown of datasets in the repository by study type, organism, diagnosis, tissue type, PCR target, which includes T-cell receptors encoded by the TRA and TRB gene loci, and B cell receptors encoded by the IgH, IgK, and IgL loci, and the PCR substrate, which includes DNA or RNA. From here, you'll want to click on Browse Repertoire Metadata. The most efficient way to search for COVID data is to simply type COVID-19 into the full text search. It may take a few moments to load your results. This query should bring up 3,414 repertoires. Note that this number can change as more data is added to the gateway. You can search via filters for study, subject, or sample. As an example, let's say we wanted to find a longitudinal study with only those diagnosed with COVID-19. We would filter for the study type longitudinal study and subject diagnosis by COVID-19. Apply filters. As you can see, this search yields 84 repertoires with B-cell data only as indicated by IgH in the column labeled PCR target. Now that you are familiar with the general query, we'll show you how we queried the data you are provided for this App-a-thon, which includes cases from the COVID-19 ISB and COVID-19 adaptive datasets. We chose those datasets because they not only contain VDJ calls and the CDR3 junction, but also express sequences. This additional information allows the CI participants to design impactful tools, for example, to harmonize TCR express sequences from differing alignment and/or assembly pipelines. In order to obtain the ISB dataset comprised of T-cell receptor beta repertoires, simply again enter COVID-19 into the full text field and then ISB under study title. Apply filters. And this query should yield 157 repertoires of COVID-19 patients with clinical data at multiple time points. To obtain the adaptive dataset, also comprised of T-cell receptor beta repertoires, remove ISB from the study title and enter adaptive. This will yield 160 repertoires, but notice here that some do not have a diagnosis. To filter down to an appropriate data set, filter by subject, diagnosis, and choose COVID-19. And now you'll have 126 T-cell repertoires. So now that you know how to query data, we'll show you how to download study metadata and annotated repertoires. The full metadata for a query may be downloaded by a TSV file by clicking the TSV button to the right of the screen. Annotated sequence repertoires are also downloadable by clicking the blue button all the way to the right of the screen that indicates Browse Sequences from 126 repertoires. You will now see a screen with a preview of the annotated sequences and a button all the way to the right, allowing a download of all sequences. Note that downloading sequence data may take a few hours. The dataset for this App-a-thon is provided to you in two files, which includes metadata and annotated repertoires. Hopefully after this introduction, those of you participating in the CI track will have a better idea as to the steps required to query additional datasets from the iReceptor gateway. We'll conclude this video by doing a walkthrough of the provided dataset for this App-a-thon. As I mentioned, the overall dataset actually consists of two merged COVID-19 studies obtained from the iReceptor gateway. There will be a total of 283 T-cell repertoires from COVID-19 cases. Because this data contains expressed sequences, these files are large, and therefore we kept the metadata separate from the annotated sequence repertoires. First, let's take a look at the metadata file. Note that a subject C subject ID column, may have more than one repertoire if samples were taken at multiple time points. As you scroll to the right, you'll see relevant clinical data such as age, race, disease status, disease length, medical history, and others. You'll note that not all subjects will have data for all of these clinical attributes. Now let's focus our attention on the repertoire ID, which is unique for each repertoire. Note that a repertoire, in the biological sense, consists of the full complement of BCRs and TCRs of an individual. However, for this schema, the repertoire is an abstract organizational unit of analysis that is defined by the researcher and consists of study metadata, subject metadata, sample metadata, cell processing metadata, nucleic acid processing metadata, sequencing run metadata, a set of raw sequence files, data processing metadata, and a set of rearrangements. The repertoire ID gathers all of this information together into a composite object which can be easily accessed by computer programs for data entry, analysis, and visualization. Even in the case that a subject has multiple data points over time, their repertoire ID will be unique for each data point. In addition to providing a unique identifier for each repertoire, it is a common variable between the metadata file and the annotated sequence file that you will use to merge on. Check the About section of this video for more information about repertoire IDs if interested. Before we examine the annotated sequence file, let's quickly define some terms. The terms are locus, gene, and allele, and we'll use visual representation. To help orient you to a gene locus, we show here homologous chromosomes, which are two pieces of DNA which carry the same genes, one inherited from each parent. A locus is the specific physical location of a gene on a chromosome, sort of like a genetic street address, if you will. Some genes have different forms termed alleles. Here we see the gene for hair color, where the individual inherited two different forms of the gene from their mother and father. There may be several different alleles or forms of a gene, and different alleles of a gene may be potentially associated with a disease or health outcome. Now finally, let's move on to the sequence data, now that you have a frame of reference for the terminology. From left to right, I'll point out columns relevant to your app creation. The locus will be TRB, indicating T-cell receptor beta for all the provided data. There are columns associated with this locus labeled VCOL, DCOL, and JCOL. These letters refer to the variable, diversity, and joining segments. During B-cell and T-cell development, a rearrangement at the DNA level joins a D segment with a J segment. The fused DJ segment is then joined to a V segment. So for example, the V call of the TRB locus is a specific V gene followed by an asterisk and then the allele. The junction refers to the complementarity determining regions, or CDRs, which are the most variable regions on antibodies and T-cell receptors generated by T and B cells respectively, where molecules bind to their specific antigens. What you are seeing in the junction AA column is the amino acid sequence of the junction. Recall that DNA is translated into amino acids which combines to form proteins. If you are participating in the data scientist track, you will be required to use the locus, VCOL, DCOL, JCOL, as well as the junction amino acid sequence from the annotated sequence file when creating your app. If you are familiar with the express sequence data and would like to use it for your analysis, You are welcome to, but it is only a requirement of the computational immunologist track. This concludes our data walkthrough. If you have additional questions, please post them on the App-a-thon discussion board, linked through the challenge info site, and they will be answered by the Precision FDA Challenge team. Thank you and best of luck.